Now, let me discuss about the sympathetic nervous system, which is a part of the autonomic nervous system. Remember, in this part of the autonomic nervous system, that is in sympathetic nervous system, noradrenaline is the neurotransmitter at most of the sites. So what you have to remember is noradrenaline is the neurotransmitter at most of the sites. Now, let me discuss how this noradrenaline is synthesized, right? It is synthesized within the sympathetic nerve terminal or in the sympathetic neuron. Let me tell you how is this synthesized. For the synthesis of this noradrenaline, we require an amino acid which is called as tyrosine. This particular tyrosine, which is present within the circulation, it is transported into the neuronal cytoplasm, right? It enters into from the circulation, right? That is from the blood vessel, that is in the circulation. Within the blood vessel, this tyrosine, it enters into the neuronal cytoplasm. Now, so this is the base material or the raw material for the synthesis of the noradrenaline. Now this tyrosine from the circulation, it enters into the neuronal cytoplasm. Now here this particular amino acid tyrosine in the neuronal cytoplasm, it gets hydroxylated, right? It will undergo hydroxylation, right? It will undergo hydroxylation. So it is hydroxylated to form what is called as levodopa, right? It is hydroxylated to form what is called levodopa. This particular levodopa is also called dihydroxyphenylalanine, right? Dihydroxyphenylalanine. Now, this is a rate limiting step, right? This is a rate limiting step in the synthesis of noradrenaline. Now this process of hydroxylation will undergo by an enzyme which is called as tyrosine hydroxylase, right? So the enzyme here is tyrosine hydroxylase. Now in the presence of this tyrosine hydroxylase, the tyrosine is converted into levodopa, which is also called dihydroxyphenylalanine. Now this tyrosine hydroxylase can be inhibited by a substance which is called as meterosin, right? So we have a substance which is called meterosin. This particular substance can inhibit the rate limiting step which is catalyzed by the enzyme tyrosine hydroxylase. Now, there is one therapeutic use of this meterosin. We have a clinical condition which is called as the pheochromocytoma, right? We have a clinical condition which is called pheochromocytoma. Now, what is this pheochromocytoma? It is the tumor of adrenal medulla where there is excessive production of noradrenaline and as well as adrenaline. Okay, right? Pheochromocytoma, it is the tumor which is originating from the chromaffin cells of the adrenal medulla where there is excess production of the noradrenaline and adrenaline. Now, when you are, the treatment for that pheochromocytoma is the surgical removal of the tumor. In that surgical removal, we use this meteorosin, right? This meteorosin can be used to control the discharge of catecholamines, right? Your noradrenaline and adrenaline, they are called as catecholamines. Remember this point, noradrenaline and adrenaline, they are called catecholamines. So this meteorosin, it is used to control the discharge of catecholamines during surgical removal of the tumor in patients with pheochromocytoma, right? Remember this point. Next, that is the therapeutic use of meteorosin. You take this levodopa. Levodopa, it is converted to dopamine, right? It is converted into dopamine. Now, let me tell you how is this converted into dopamine. Levodopa, it is converted to dopamine by the action of a non-specific decarboxylase enzyme, right? In the presence of enzyme decarboxylase, this levodopa is converted into dopamine, right? So the enzyme here is decarboxylase, right? This is a decarboxylation step. So in the presence of enzyme decarboxylase, the levodopa is converted into dopamine. Now, you take this decarboxylase enzyme. This particular decarboxylase enzyme can be inhibited by two important drugs, right? So the drugs what we have which can inhibit, right, which can inhibit the decarboxylase is, we have carbidopa, right? We have carbidopa, and we have another important drug, that is benserazide, right? Another important drug which is called benserazide. So carbidopa and as well as benserazide, they will inhibit the decarboxylase, thereby the levodopa is not converted into dopamine. All right? Now, so in the process of the synthesis of your noradrenaline, you see what is happening? Levodopa is converted into dopamine. Now this particular dopamine, where is it stored? Remember, dopamine, it is transported to the storage vesicles within the sympathetic neuron. This particular dopamine, it is transported to the storage vesicles. Okay, so now this is transported into storage vesicles within the neuron, within the sympathetic neuron. Now the transport of dopamine into the storage vesicles can be inhibited by a drug called reserpine. So we have a drug called reserpine. This particular reserpine will inhibit, right? This particular drug reserpine will inhibit the transport of dopamine to the storage vesicles. All right? Next, what is the next step from the dopamine? Remember, the next step from the dopamine is, this dopamine is converted into your noradrenaline in the presence of an enzyme which is called dopamine beta hydroxylase, right? It is converted into noradrenaline. So you have to remember a very important point, that all these entire steps, they are taking place within the sympathetic neuron. Okay, now, so this dopamine in the presence of an enzyme which is called as dopamine, right? In the presence of an enzyme which is called dopamine beta hydroxylase, this dopamine is converted into noradrenaline. right? This dopamine is converted into noradrenaline. Now, this particular enzyme, this dopamine beta hydroxylase, is inhibited by a substance which is called as disulfiram. Okay, so this can be inhibited by disulfiram, right? Remember this point. So at each level, what I am trying to tell you is the enzymes which are useful for the conversion, and I am also trying to tell you what are the substances which can inhibit that enzymes. Now your noradrenaline is formed. Now this particular action of the noradrenaline can be terminated by two ways. The action of the noradrenaline can be terminated by two ways. So if you take this noradrenaline here, the action of the noradrenaline can be terminated by two ways. One mechanism how noradrenaline action can be terminated is, it is mainly by the reuptake in the vesicles. Now what will happen in the storage vesicles? The dopamine, in the presence of dopamine beta hydroxylase, is converted into noradrenaline. Now whenever the sympathetic neuron receives the stimulus, this noradrenaline is released, right? Noradrenaline is released from the nerve terminal. Now the action of this noradrenaline is terminated by two ways. Mainly the action of the noradrenaline is terminated by reuptake in the vesicles, right? Right? So the action of the noradrenaline, it is terminated mainly by reuptake within the vesicles. Now even this particular reuptake can also be inhibited by certain drugs. Remember, this particular reuptake can be inhibited by, number one, cocaine, and the other drug is tricyclic antidepressants. So cocaine and as well as tricyclic antidepressants, they will inhibit the reuptake into the vesicles. Next, the other way how the action of the noradrenaline can be terminated is by the metabolism, right? It is metabolized by two important substances, that is, right, the action is also terminated by metabolism. Now you take this particular metabolism. The metabolism is done by monoamino oxidase, wherein the action of the noradrenaline can be terminated. The other way how the noradrenaline action can be terminated is by the another important enzyme, that is COMT. If you take this COMT, this is catechol O methyl transferase, right? In the presence of these two enzymes, the noradrenaline will be metabolized and the action of the noradrenaline can be terminated. Next, now you see here we have another important catecholamine which is called adrenaline. Now where is this adrenaline formed? Adrenaline, it is formed in the adrenal medulla. What is required for the formation of the adrenaline? Remember, right? Remember this point. Noradrenaline is converted into adrenaline in the adrenal medulla. Right? It occurs within the adrenal medulla. Now, what is this particular step? This conversion of noradrenaline to adrenaline is the methylation step, right? By the process of methylation, the noradrenaline is converted into adrenaline. Okay, so this methylation step, it occurs in the cytoplasm with the help of an enzyme which is called as transferase. Now what is this particular transferase? That is the phenylethanolamine N-methyl transferase. Now let me discuss about that methylation, but before that let me shortly revise like what we have discussed until now. In the sympathetic nervous system, at majority of the sites, we have the noradrenaline as a neurotransmitter. Now what I am trying to tell you is, how is this particular noradrenaline synthesized or formed? The amino acid which is required for the synthesis of the noradrenaline is a tyrosine. This particular tyrosine, from the circulation, it is taken up into the neuronal cytoplasm. Now this tyrosine, it will undergo hydroxylation in the presence of tyrosine hydroxylase to levodopa, right, which is also called dihydroxyphenylalanine. This levodopa, in the presence of enzyme decarboxylase, is converted into dopamine. Now this dopamine is sent into the storage vesicles. Within the storage vesicles, in the presence of an enzyme which is called dopamine beta hydroxylase, this dopamine is converted into noradrenaline. Now, once the sympathetic neuron receives the stimulus, the noradrenaline is released as a neurotransmitter. Now, at various levels, the steps can be inhibited. Now, for example, you take the hydroxylation step by tyrosine hydroxylase. This meteorosin is used during the surgical removal of the pheochromocytoma. Next, you take this decarboxylation step. This particular decarboxylase can be inhibited by drugs, which is called carbidopa and as well as benserazide. Next, the dopamine whichever is formed is sent into the storage vesicles. The dopamine sending into the storage vesicles can be inhibited by reserpine, right? Can be inhibited by reserpine. Next, the another important thing is this dopamine beta hydroxylase can be inhibited by a substance which is called disulfiram. All right. Now, the noradrenaline whichever is being formed, the action is terminated via two ways. Mainly, the action is being terminated by the reuptake within the vesicles. Even this particular reuptake can be inhibited by drugs called cocaine and as well as tricyclic antidepressants. And the other way how the action of the noradrenaline can be terminated is by the metabolism, by monoamino oxidase, by catechol O-methyl transferase enzyme. Now, within the adrenal medulla, the noradrenaline is converted into adrenaline by a process of methylation. Now, for this particular process of methylation, we require an enzyme which is called phenylethanolamine N-methyl transferase. Now, let me discuss about that particular enzyme, that is phenylethanolamine N-methyl transferase. So, now you take this particular noradrenaline, it is converted into the adrenaline, right? Noradrenaline is converted into adrenaline where in the adrenal medulla. So in the adrenal medulla, noradrenaline is converted into the adrenaline, and that particular process is called as methylation, right? That particular process is called methylation. Now, this particular methylation will occur in the presence of an enzyme which is called as phenylethanolamine N-methyl transferase, right? In the presence of the phenylethanolamine N-methyl N-methyl transferase. Okay, so in the presence of this particular enzyme, process of methylation will take place. The point what you should remember here, you take the sympathetic neurons. The sympathetic neurons, right, they lack this enzyme, right? Sympathetic neurons, they don't have this enzyme. This enzyme is present only within the adrenal medulla. right, wherein the noradrenaline can be converted into adrenaline. Okay, so the sympathetic neurons, they lack this, the phenylethanolamine N-methyl transferase. So therefore what you have to remember is the catecholamine synthesis in the sympathetic neuron within the vesicles, the synthesis is stopped at the noradrenaline level, right? So that is the reason why in sympathetic neuron the synthesis stops at noradrenaline level, right? The synthesis stops at noradrenaline level within the vesicles of the sympathetic neurons. Now, once these particular sympathetic neurons are stimulated, right? Once these particular sympathetic neurons are stimulated, stimulation of these neurons by action potential will increase the influx of the calcium, and that will result in the exocytosis of noradrenaline in the synaptic cleft, right? So whenever these sympathetic neurons, whenever they are being stimulated, what will happen? There will be influx of the calcium within the sympathetic presynaptic nerve terminal, and from this sympathetic neuron there will be exocytosis of noradrenaline. Okay, now you take this particular exocytosis, this exocytosis is also inhibited. This exocytosis is inhibited by the drugs which is called as bretylium and as well as guanethidine. So this exocytosis of the noradrenaline from the sympathetic neurons is inhibited by bretylium and as well as guanethidine, right, and as well as guanethidine. Okay, now this noradrenaline, whichever is released in the synapse, what it will do, this particular noradrenaline, it will act on the postsynaptic receptors, right? It acts on the postsynaptic receptors. By acting on, right, by acting on the postsynaptic receptors, it will have the various effects. Right, it will have the various effects. Now this noradrenaline, whichever is released from the synaptic nerve terminal, it will also act on the presynaptic receptors. Right, it will also act on the presynaptic receptors. Now by acting on the presynaptic receptors, the noradrenaline will modulate its own release. Right, it will modulate its own release. Okay, so that is what is your noradrenaline doing. So noradrenaline, whichever is released from the synaptic nerve terminal or from the sympathetic nerve, it acts on the postsynaptic receptors, thereby it will have the various effects. It acts on the presynaptic receptors, thereby it will modulate its own release.